starting to record, and I'm starting to share the screen and present today's lecture. So, as we discussed many times, one of the major motivation in molecular imaging in clinics is the identification of patients who express a particular molecular target in the tumor, and then they would most likely benefit from targeted therapy. And the most straightforward way is, of course, to radio-label therapeutic monoclonal antibody. So one could follow its fate in vivo and observe if a monoclonal antibody accumulates in a tumor site. So it's quite likely that it's because of specific interaction with the target, and this patient most likely would benefit from therapy. If not, well, then it's probably low expression of molecular target, and we need to look for an alternative way of treatment. However, antibodies are excellent therapeutic agents. They stay long in circulation, they are very specific, and generally speaking, you have a lot of time to reach a target. For imaging, it's not really an advantage. So antibodies are very big, 150 kilodalton. That means that they extravasate, so penetrate from the other tissues quite slow, because pores in the vessels are typically much smaller. Also, they clear slowly from normal tissues. So therefore, the background remains to be high, and it's difficult to see a tumor. You could appreciate this image. So one could visualize the black dots, which symbolize a tumor or manifest as a tumor, but apparently you need arrows to understand that this is a tumor. It's quite a high background, and one could see also activity in major blood vessels. But this is only a part of the problem. The problem is that antibody can accumulate in tumors unspecifically, independent on their molecular target, due to so-called enhanced permeability and retention, EPR effect. So then the imaging would be deceptive, because we would believe that the tumor is target positive while it's target negative. So overall, it's not really facile procedure when imaging has an optimal time at 5 days after injection, between 4 and 7 days, so it's slow. Contrast remains to be low, and we still might have false positive finding and treat a patient who will not benefit from therapy but would be exposed to risk of side effects. Well, the development of antibody-based imaging agent has a long history, and quite long ago, even 90s, it was understood that smaller fragments of antibody can better imaging properties. For example, if you treat antibody with enzyme pepsin or papain, the constant part of an antibody will be digested, but Fab fragment, antigen binding parts, would be preserved, and depending on the enzyme, they would be either dimeric with a molecular weight of approximately 110 kilodalton or monovalent. Monovalent variants are below the renal cutoff size, so they are filtered through glomerular membrane and kidney and excreted quite rapidly. So we gaining in this case two aspects. So the smaller is an imaging probe, the quicker is blood clearance rate, and extravasation and tissue penetration could be also improved. In early 2000, well, late 90s, smaller fragments have been developed. For example, it was so-called single-chain variable agent. So framework has been removed further, and only domains of antibodies which are responsible for molecular recognition has been arranged as a single polypeptide chain, which, if you like it, might retain affinity and specificity to a target. And that is further reduction. Probably the best what one could do with immunoglobulin is to use so-called VHH domain or nanobodies. They are derived mainly from camelids. So camel, and interestingly, the sharks, the fish, have also very interesting antibody which contain only heavy chain but not light chain. So the target binding domain is even smaller, just 18 to 15 kilodalton, and it's smaller, and it demonstrates quite impressive imaging, and it's specific because the border for EPR and specific accumulation is around 45 kilodaltons. Moreover, if you use peptides, which are typically derived from peptide ligand of receptors that are overexpressed in tumor, you can get absolutely fantastic images with very high contrast. So I would say that development of small polypeptide is the way to develop a good imaging probes. The problem is that not all targets are receptors. So carcina embryonic antigen TAG72, they are just antigen. They have no target, the receptor nature. More, not even receptor have identified peptide ligand. Well, for example, HER2, very popular target for anti-cancer therapy, does not have any ligand at all. Not identified despite 30 years of active search. So would be desirable to develop small peptides which can bind to molecular targets in cancer. The discovery of phage display deserved the Nobel Prize in 2018, and George Smith deserved for development of so-called phage display technology. And this potentially enables selection of quite small binders to any thinkable target. So nowadays, I think everyone is an expert in viruses, and we know that normally viruses just hijack DNA machinery of cell to replicate first the DNA and to replicate protein of capsid envelopes. So what George suggested is to make a double hijacking. So to use or to cheat phage viruses. So what he did, he made by synthesis the strains, the random sequences of DNA encoding peptide and built into viral DNA. And by random synthesis, you might create a library containing millions or hundreds of millions of different variants. And in this case, the phages expressed the desirable or different peptide on the capsid. So the cycle was arranged like that. So a library of phages expressing different peptides has been generated and exposed to antigen which was immobilized, for example, on magnetic beads. So if the library is big enough, stochastically, there might be a variant which bind to molecular target. So after exposure, you let your binder to bind to molecular target on the beads, wash away what is not bound, so you will have enriched smaller library which is capable to bind to molecular target. Then you elute them, and then you just amplify in E. coli or whatever host you have for this phage. You create a much bigger library, and you repeat the cycle in this way. But in this case, you increase in harshness of washing conditions, and only stronger binders would remain bound to molecular target. And this process is called panning, like washing the gold from the sand. And if you have several panning sites, you can select the very strong binders and use it for targeting. Greg Winter, who was another Nobel Prize winner, used this process for further development of antibodies, for example. Problem is that the short peptides which have been selected using such libraries had a quite poor binding strength, poor affinity to molecular target. That sounds strange because, in fact, the phages which carry those peptides were bound quite strong to target. However, there is sort of pre-organization of peptide on surface of the virus. So peptide is restrained in its movement, in its conformation, and during display, its certain conformation of a peptide has a high binding strength, high affinity. If you just cut the peptide from the phage, there will be a variety of different conformations, so you need pre-organize somehow peptide for binding, and organization means the loss of entropy, and in the end, loss of Gibbs energy, and Gibbs energy is connected with affinity directly. So, to overcome this issue, a scaffold concept has been suggested. And scaffold contain a robust domain which folds spontaneously. So one could find in a Mother Nature different scaffold which you needn't pre-organize, you needn't even add the disulfide bridges, just form spontaneously. One of example, by the way, is of course immunoglobulin, which is example of the scaffold. But scaffold protein contains this robust framework scaffold, which holds in very defined position the variable amino acids of the library. So in this way, you have a big library which has very well defined geometry or stereometry of the folding. And that enables really high affinity binder selection. So in the end, you have a variety of different scaffolds, which has nice advantages for using as imaging probes. First of all, they're quite small. The best variant is only 3 kilodalton. Well, and then typically the size varies between 3 and 20 kilodalton. And this small size enable rapid extravasation. They just filter through the pores in the vessels rapidly outside the blood flow. They diffuse rapidly in extracellular space, so they have a good chance find the antigen and bind it. And in the end, what is unbound clears from the blood flow and reduces background. They could be selected for very high affinity. And when I will talk about affinity, I may have to make reservation. So the word affinity means the strength of binding, but there are misuse. So I will use the word affinity as synonymous as of dissociation constant at equilibrium. And the lower is a value of dissociation constant, the stronger is a binding. So typically, scaffold proteins have affinity from a few picomole to 10 nanomole. And for comparison, good therapeutic antibody have affinity in the same range. What is important that they could be produced using peptide synthesis, which is a very cheap way to produce them, or they could be produced in prokaryotic host recombinantly, which is big advantage in comparison with monoclonal antibodies, which are usually produced using mammalian hosts. And it's much more difficult, provides low yield, and increases the cost. So we could produce scaffold proteins much cheaper than produce antibody. And by gene engineering, you could relatively easily create multimeric construct, multivalent construct, fusion protein, chimeras where protein is genetically fused with, for example, peptide amino acids and so on. So now it's a rather hot area in just big pharma. Several scaffold proteins have been evaluated as an imaging probe. And here is a cartoon showing a relative size to antibody. We would talk today about Affibodies, DARPins, and adapts, and you could see that they are really smaller than an antibody, which offers advantages in human targeting. It happened that the first class of scaffold protein, which has been evaluated for radionuclide imaging, was Affibody molecules. And invention of Affibody has been done by Perocchi Nygren, who is now professor of biotechnology at Royal Institute of Technology in Stockholm, and that time he was a young postdoc, trying to find a solution for problem he encountered with development of antibodies. And preclinical development was performed by Professor Anna Arlova, who read an introduction lecture. So, what did Perocchi? He used one of the mains of protein A from Staphylococci. So, Staphylococci tried to protect against immune system of the host by producing on the surface or expressing protein A, which has a capability to bind host immunoglobulin. In this way, the surface of this bacteria is coated with outer layer of the host antibodies. And of course, it's difficult to recognize them. So the protein A consists of several domains, and one of them, B domain, has been used. It's a steady construct which contains 3 alpha helices, and helix 1 and helix 2 are responsible for binding to constant region of immunoglobulin, and helix 3 just is there to provide robustness of the structure. So what did Perocchi? He has randomized 13 amino acids on protein A and B, and that were exactly those positions which are responsible for binding to immunoglobulin. So affinity to immunoglobulin has been raised in this way, and a library has been created which enable selection of variety of different Affibody molecules with different specificity. The latest size of the library was 20 billionth variant, and well, I've seen the data that whole repertoire of human antibodies contains 100 billion, so it's comparable in the immunoglobulin repertoire of humans. We work quite a lot with Affibody, so it happens that me along with Professor Orlova were first who made the labeling and in vivo characterization. I think this is a very informative slide which compares imaging of anti-HER2 Affibody with imaging using anti-HER2 antibody trastuzumab, which is commonly used in clinic and workhorse in the treatment of breast cancer. And trastuzumab accumulated in tumor already 6 hours after injection. Still, the activity in blood pool was high. One could see the activity in the heart here and in carotids, and also high activity in liver, and taking into account that, for example, liver is very common metastatic site. It was far too early to make an image. So even 24 hours, the contrast was not good, and in mice, it requires 3 days to produce image with reasonable contrast. With Affibody, we managed to do it with 6 hours after injection. Recently, we made a comparative evaluation of Affibody molecules and FAP2 fragment for imaging of carbonic anhydrase 9, which is an antigen expressed in renal cell carcinomas. And FAP2 is probably one of the best forms of immunoglobulin-based imaging probe. Still, one could see that 4 hours after injection, the contrast between tumor and the rest of the body was not really high. And there was a difference, clear difference, with Affibody molecules where there was an excellent contrast. So only kidney were shiny, but there was no accumulation in the body. Even 24 hours contrast has been improved for FAP2 fragments, but still Affibody molecules were better. And high contrast determines sensitivity. But what is important that for antibodies, there is always some specific accumulation. This is murine model where a team of Riley from Toronto made imaging, and one could see that although there is a specific accumulation, unspecific accumulation in tumors was quite high. And in clinics, it might confuse the clinicians. A diagnosis might be false positive. With Affibody molecules, we have shown that specific variant provides high and bright imaging, but unspecific has no accumulation in tumor at all. So we just superimposed the contour of mice. And the uptake in non-specific uptake in tumor was like in muscle, roughly 100% lower. So the chances for false positive is pretty low. Of course, the labeling chemistry is essential, and during our previous lectures, we have considered different approaches, and I hope that me and Angelica managed to convince you that by influence target binding properties of protein, cellular processing of the tracer, the biodistribution, labeling chemistry might make a big difference and in the development of imaging probes and make a difference between success or failure. So as we discussed earlier, very important aspect is consider residualizing or non-residualizing label. Roughly speaking, it's metal versus non-metal label. And generally speaking, it depends a lot on the internalization rate. So if internalization rate is low, the use of non-residualizing labels is justified. It happened that the binding of nearly all scaffold proteins we studied was very, very slow. So this is experiment on cellular retention and internalization of cells, and we could see that internalized activity is about 20, maximum 25%. And this is essentially independent on target or antibody molecules. There are very few exceptions for that. And that means that the retention is independent on internalization, intracellular retention of the metals, and we could use non-residualizing label. And we still can get good image of the tumor, but not, for example, kidneys, where antibody molecules are reabsorbed and create a very high background in the lumbar area. We have discussed earlier that adjusting residualizing properties of the label and overall physical chemical properties of the label, we could suppress, for example, hepatobiliary excretion, suppress retention in kidney, and end up in high contrast imaging. For example, using cysteine or mercaptoacetyl based peptide chelators. We also have discussed that one could, for example, avoid hepatic uptake using right chemistry and enable imaging tumor while keeping liver uptake low, unlike wrong chelating moiety where activity in liver is much higher than in tumor. And optimizing labeling chemistry was a long journey, and pursuing an optimal label, we use more than a dozen of different nuclides, and once upon a time I counted that we use around 100 different labeling methods to find the best one. So, but it paid off. There are certain aspects in targeting using scaffold proteins which are not connected to labeling chemistry, but still important. Our problem was that before us, no one worked with scaffold protein at all. So all antibody or immunoglobulin-based imaging probes were several-fold, at least several-fold bigger than scaffold proteins, and typical peptide-based probe was at least several-fold smaller than scaffold protein. So it was completely unclear what from previous knowledge we can translate to our scaffold proteins. So one question we had to clarify is influence of affinity. So if we have rapid internalization and if we have residualizing label, so activity is internalized together with a target trapped inside, and essentially retention of activity starts to be independent on affinity. So because we switching to irreversible process of internalization. However, if we have slow internalization, then we have reversible process. There is a possibility that Affi-body molecules would dissociate from the surface of cancer cell. And then we need to know to which one level we have to raise affinity to provide the good targeting. So to clarify this issue, we performed experiment with anti-HER2 Affi-body molecules. And we had one variant with very good affinity, 116 picamole. And second was not bad. It was 4 nanomoles, but it's more than order of magnitude weaker binding. And we compared them in mice bearing human xenograft SKOV3, which has approximately 1.2 million receptor per cell, and LS1740, which has approximately 40,000 receptor per cell. So we took a whole scale, and we injected 1 microgram of Affi-body molecules radio-labeled and compared the biodistribution at 4 and 24 hours after injection. What we have found that in a case of high expression, it doesn't matter if affinity is 120 picamole or is it 4 nanomole. The tumor uptake was the same. So, however, if we had longer time, the low affinity Affi-body molecules were cleared from the tumor, and tumor uptake was lower, while high affinity remained on the same level. The situation was completely different if we had xenografts with low expression of HER2. First and foremost, in this case, absolute value of uptake was lower even 4 hours after injection, but the difference was very big between high affinity and low affinity antibody molecules. Of course, retention was weaker even for high affinity variant at 24 hours after injection, but the difference is just further increased between the high and low affinity. So we concluded from this study that low nanomolar affinity is good enough for imaging of molecular targets with high expression level, around 1 million receptor per cell. This is level of expression of HER2, level of expression on epidermal growth factor receptor, and some other molecular targets. If we need to image targets with low expression for 40,000 receptor per cell, we need to have subnanomolar affinity in picomolar range. And there are quite abundant targets which has that level of expression. For example, HER3 or insulin-like 1 growth factor receptor, which are responsible, for example, for resistance to trastuzumab. So now we understood approximately to what level we have to develop our agents. Well, how to improve affinity? So we know how to do. And there are generally two approaches. One is pretty straightforward. So you dimerize your binder. In this way, you gain so-called avidity effect, and typically you reduce dissociation rate with dimerization and accordingly increase affinity by order of magnitude. And it's relatively straightforward process. Good master student could do this work within 1 month or 2. Alternative is process so-called affinity maturation. This is appreciably trickier. So you generate several clones of antibody molecules or whatever binder you have. You identify amino acids which are concordantly in certain position. So you create a second library where those positions are fixed with benign amino acids, and you make a second round of panning, and not seldom is 3 or 4 steps of affinity maturation. So this is a more complicated way, and good researcher with PhD degree could do it 6-7 months. So of course dimerization was attractive. However, it turned out that this way is not really good. So when we start work with antibody molecules, the first variant we have selected, clone 4, has affinity of 50 nanomole. And that was not really good. So to overcome this problem, we created dimer, and the dimerization per se worked very well. We had more than order of magnitude improvement of affinity. However, it turned out that the dimeric variant had no better tumor uptake and no better retention compared to monomer. 24 hours, yeah, we had advantage of dimeric form. However, when we got first affinity maturated variant of Fib body molecules with affinity of 22 picomole, it beyond any reasonable doubt had much better tumor uptake and much better tumor to blood ratio, rising the imaging contrast and potential sensitivity. More interestingly, that when we did dimer of affinity maturated variant, again, the tumor uptake was for dimer lower and compared with monomer. So the hypothesis was that dimerization results in at least doubling of molecular weight, of course, and doubling of the size of Fib molecules, and that prevents the tumor accumulation. So besides our work, several other groups worked with Fib molecules with other clone and label with two different nuclides with fluorine 18 or copper 64, and independent on the label. They had again worse accumulation in tumors compared to monomeric form, although every time the affinity of dimer was better. So, based on our and other research, we dare to state that, well, affinity maturation increases both uptake and retention of radionuclide in a tumor. So, if we use alternative dimerization, it increases retention by decreasing off rate or dissociation rate, but however, it decreases tumor uptake, and most likely negative influence of dimerization on tumor is associated with bigger size of dimeric form. We further on have confirmed it with other scaffolds. So we found that affinity maturation is the preferable way to increase affinity compared to dimerization. So next step would be to discuss clinics and to discuss other scaffolds. Should we make a short break? Hello?
Yes.
Okay. So my suggestion that we return in 10 minutes and move a little. Okay. Okay. Hello. Could those who returned from the break switch on videos to mark that you are back? Well, then we probably need to wait a little bit more.
Некоторые пишут в чат, что появились.
Ну, проблема, я не вижу одновременно и presentation screen и чат. Могу я еще раз попросить мигнуть так, видео? Ладно, дадим еще 30 секунд. И, наверное, надо будет начинать все равно. Ладно, есть преимущество определенное. Виталина, Наталья, Александр, Катерина, Мария Третьякова. Ну ладно. Будем надеяться, что остальные подойдут. Хорошо. Then we're switching to English back and continue our talk. Well, of course, what I was talking about was mainly about preclinical imaging. And step to clinics is always a very big. We remember that not more than 10% of all agents or therapeutics which enter clinical trial pass it through the product. Human being is definitely not 70-kilo mouse, and there is a different target expression, different physiology, different heart rate, and so on. So it might be very different compared to mice. However, Affibody molecule passed this exam very nicely. So there was a phase 1/2 clinical trial of imaging of Affibody molecules using indium-111 as a label and SPECT as a detection technique. And we have visualized a variety of targets. Essentially, sensitivity was 100%. We were able to visualize bone metastasis, brain metastasis. It's very often a challenge because brain is hidden behind blood-brain barrier. However, in tumors, it's often disrupted, and we managed to visualize HER2 expressing metastasis in breast cancer. We were successful in imaging of liver metastasis, and this adrenal metastasis was very important for us. So if you remember, we had always quite high renal retention of activity in the case of residualizing metal label. And we worried actually that even imaging of bone metastasis close to kidney would be obscured by so-called reconstruction artifact when we have high activity in kidney. However, this adrenal metastasis is sitting right on the top of a kidney. Still, it has been visualized. The next step was development of imaging probe for PET. And positron emission tomography is advantageous technique, superior, providing better quantification, better spatial resolution, better temporal resolution. And we have shown that Affibody molecules can visualize HER2 positive tumors shortly after injection. So this cartoon contains images pairwise of the same patient, showing first FDG image, fluorodeoxyglucose, which identify tumor site, but doesn't say if there is HER2 expression or not. So in case of HER2 negative patient, there were dozens of metastases in the patient, but not single one has been visualized using anti-HER2 Affibody molecules. On the opposite, if we had HER2 positive cancer, we had exactly the same position as with FDG visualized using Affibody molecule, including bone metastasis, liver metastasis. But we also were able to visualize certain metastases which were not really seen using fluorodeoxyglucose. That was a moment of triumph, I would say. First and foremost, the word HER2 negative, it's deceptive. In fact, HER2 negative breast cancer could express up to 500,000 receptor compared to HER2 positive, it's probably order of magnitude higher, 3-5 million receptor. But we managed in this case to select these conditions where we could discriminate between HER2 negative and HER2 positive patients. And second lesson was that now, okay, PowerPoint has issues. I try to restart. Okay. Sorry about this. So we managed to get a good and high contrast imaging. You could here compare imaging using clinical imaging using antibody molecules and antibodies. And here is zirconium, it's a mistake. Zirconium labeled antibody which was used for visualization of HER2 expression 5 days after injection. Here is gallium-labeled antibody, which was used for imaging only 4 hours after injection. I think there is no need in many comments. You can yourself compare the contrast of images. So the development of HER2 antibody was a story of success. Currently, we are working on several other antibody-based imaging probe besides HER2. For example, it's EGFR or HER1. It's a molecular target for antibodies panitumumab or cetuximab, and is molecular target for in head and neck cancer or colorectal cancer. CA9 is interesting target because it first is a target in renal cell carcinoma, but it also is overexpressed in hypoxic tumor. And when tumor is hypoxic, it's more radio resistant, it's more drug resistant. So if you're sure that you have hypoxia in a tumor, you need to reconsider your treatment regimen. The insulin-like growth factor 1 receptor or HER3 are major suspect or probably even major culprit in trastuzumab independence. So if you have HER2 expressing breast cancer and you treat it with trastuzumab, patient for a year or two respond nicely to therapy, and tumor growth rate is quite suppressed. However, after a while, clonal selection happens in the tumors, and they start to express either IGF-1R or HER3 and switch mitotic signaling to those targets. And if we can visualize the expression of the target, it would be a strong signal for us that we need to switch our therapy to something else, not just blocking trastuzumab signaling. And PDGFR is a platelet-derived growth factor receptor, is a target in tumor stroma. We learned quite a lot about tumor biology, and if earlier we considered a true tumor mainly like bag with malignant cells, it turns out that it's not completely true. There are plenty of non-malignant cells, normal cells in a tumor. In lymphomas, for example, the malignant cells are just majority. But tumor stroma functionally support malignant cells, and if we can knock stroma out, you will destroy a tumor. So platelet-derived growth factor receptor is considered as promising molecular target for therapy of cancer. Well, not every image is as beautiful as image of HER2, but we have to admit that we are only in the beginning of our way, and putting enough efforts, we can improve appreciably the imaging using any antibody molecules. Here is just two examples. It's the first image we got in mice of expression of epidermal growth factor receptors. We were capable to image a tumor, yeah, but of course there were high kidney, it was radiometal indium label, but there was a plenty of activity in liver besides and in gastrointestinal tract. After 7 years of hard work, we managed to come to this level where, of course, kidney remains in place, but you could see we nearly completely suppressed the accumulation in liver and suppressed the uptake in gastrointestinal tract while keeping the tumor well imageable. The same is with HER3. Again, the very first image. Do you see me? Because, okay, it's blinking. So you could see the tumor was visualized, but again, kidney, liver, gastrointestinal tract, a lot of radioactivity uptake, and well, essentially would be very difficult to image a tumor which is below diaphragm. Again, optimization of a label, injected dose, antibody molecules result in the situation when, besides tumor, only a liver was visualized. So it's possible to make candy from not very nice substance if you know what to do. Antibody molecules is not the only possible scaffold, and another scaffold which is now in work is ADAPT or albumin binding domain derived affinity protein. The mastermind behind development of this tracer was Sophia Huber, who is, when she is not diving, a professor in biotechnology at Royal Institute of Technology. And major work in this project did two PhD students, Sarah Lingbu from Royal Institute of Technology, who made biotechnology part, and Javad Garusi from my group, who made labeling chemistry and preclinics. So, ADAPT is based on albumin binding domain of protein G. The nature of protein G is similar to nature of protein A. So it's defense or camouflage of cancer, of streptococci against immune system. So streptococci express albumin binding domain. It catches the albumin of the host and create a coat around which complicates recognition by immune system. Of course, during the evolution of streptococci in protein G, this protein was developed to be non-immunogenic because its goal is to hide from immune system, not to expose to immune system. And ABD is pretty small. The basic core is 5.2 kilodalton, and it's non-immunoglobulin. It forms very strong, robust scaffold. We used earlier immuno-ABD for modification of pharmacokinetics of antibody molecules. So for therapy, to avoid the kidney reabsorption, we fused antibody molecules with ABD, and after injection they bound to host albumin, and were not filtered through the kidney. So right now is ongoing phase 3 clinical trial with anti-interleukin antibody molecules fused with ABD, and there are hundreds of patients now who were exposed to antibody ABD fusion biweekly for full year, 20 milligrams, and there was no immune response. It's completely non-immunogenic. And what is important for us that folding of ABD is independent and disulfide bridges, so it folds spontaneously. It denatures chemically or thermally, but it refolds with a high fidelity, which makes it very, very convenient for labeling. In principle, we can tune affinity of ABD to albumin by mutagenesis, which is important, and Sophia managed to engineer a second binding site in ABD, which is far not trivial biotechnology task because albumin is pretty bulky as such, but she managed. And she selected HER2 binder as well as a number of pretty high affinity binders to other molecular targets. Well, this cartoon shows again in the same scale the size of ADAPT and size of immunoglobulin G, most commonly used antibody, and you could see that ADAPT is much smaller. Sophia's team managed to select several adapts with low nanomolar affinity. HER2 was a champion, but was also 3 nanomolar binder to tumor necrosis factor alpha or to HER3. So to validate that it's possible to target tumors using ADAPT, I asked her to completely obliterate binding to albumin. For therapeutic application, binding to albumin is very good, but for imaging, when we need to have rapid clearance, we don't want to have any binding to albumin. So major intrigue in this study was if we manage to suppress albumin binding or not. So it turns out that we managed. All these images has been done 1 hour after injection in mice, and they were head-to-head. So they performed at the same time and in parallel. And both those mice carry the HER2 expressing xenograft. This mouse, though, was pre-injected with anti-HER2 antibody trastuzumab, and ADAPT binds to the same epitope as trastuzumab. So pre-injection of large amount of trastuzumab completely blocked receptor and prevented binding with ADAPT, but otherwise we could see that the tumor uptake was high, and the only place with a higher uptake was kidney. Well, the same problem as with Affibody, high renal reabsorption. We also managed to show that we can use it also for positron emission tomography using gallium in the label. This image has been done using clinical PET camera, and we again managed to select conditions when a SCAOV3 tumor with a high expression, clinically relevant, could be visualized clinically, while tumor with low expression were not essentially seen. As usual, the proof of principle was followed by years of work trying to understand how to use it in the best way. For example, we have shown that if we place label at C-terminus, not at N-terminus of ADAPT, we gaining twice high tumor to lung and tumor to liver ratio. And both lungs and liver are major metastatic site. So increasing the contrast towards lung and liver, we improve the sensitivity of imaging. So this work made sense. We also have shown that the use of non-residualizing iodine label shows very good tumor uptake and helps to avoid long retention in kidney. So 4 hours after injection, the kidneys were not visible in microspect imaging. We could also hope then that the same type of behavior would be in the case of radiofluorine label. And major efforts have been directed to gallium label because it permits imaging using positron emission tomography, best modality. And after several years of optimization, we managed to increase more than fourfold contrast toward liver, contrast to bone, and bone is also another important metastatic site. So we hope that it will work with positron emission tomography. We have also demonstrated that we can label adapts using 3-carbonyl technetium chemistry and use it for SPECT. The behavior was completely different. We earlier found for affibody molecules that HHH tag is better than hexahistidine tag, but that was not the case for adapts. But on the opposite, when we have a 3-carbonyl label and hexahistidine tag, we had tumor to blood ratio of 100 4 hours after injection, and very, very decent tumor to organ ratio to majority of organs, except from kidney. We have also shown that if we saturate HER2 receptor with antibody trastuzumab, we suppress binding. So we have demonstrated that the targeting is HER2 specific. Next step in clinical translation has been performed in Tomsk. And I have to give a major credit to 2 clinical researchers, to Professor Vladimir Chernov and Dr. Olga Bragina, who carry this study on their shoulder. Important role in transfer of technology played also Elena Stasiuk, so there was a good team created in Tomsk, and that permit us to put Tomsk in the front line of new tracer development. So these are clinical images using technetium label products. So the same feature as in mice, high uptake in kidney, liver was also visible, but tumors, HER2 with high level of HER2 expression, were very, very clearly visualized already 2 hours after injection. With antibody, you will see only blood pool. So you need to wait for 5 more days before getting an image, not even like that, which much worse contrast. We did, together with Olga and Vladimir, the full distribution. We evaluated several injected dose to find the right one. And we had the same problem as affibody, so that was HER2. So we had expression in HER2 negative tumors. And we found that the best contrast and best discrimination between HER2 positive and HER2 negative tumors can be obtained when injecting 500 microgram of ADAPT6, and it's already 2 hours after injection. So this diagram shows tumor to contralateral breast ratio. It's a common way to identify the clinical parameters, so you measure tumor to background. So red crosses are HER2 positive tumors, the blue ones are HER2 negative. And we could very clearly see that at any time point, the uptake of activity in HER2 positive xenografts is much higher than uptake in HER2 negative. So we have a clear cutoff value here, and 500 microgram was an optimum. So it was very good clinical study we did together. Another scaffold is DARPin. That very different in its nature. DARPin called design ankyrin repeat, and the binder is assembled of 4 or 5 homologous repeats, each containing 2 alpha helices in 1 loop. And it might be 4 or it might be 5 such repeats. And the concept belonged to Professor Andreas Plukton from Zurich School of Technology. And the DARPin project was started by academic Dev from Ovchinnikov Institute in Moscow and Professor Chernov from Tomsk, and Olga Bragina was again the clinical researcher doing the clinical imaging. Still, we had to learn how to label. Selection of nuclides in Tomsk are unfortunately a bit more restricted, and we could run only SPECT imaging. So Associate Professor Angelika Vorobyova, who holds a number of lectures, made major preclinical work in Uppsala, and we started to compare 2 labels, direct iodine or hexahistidine label. We have found that apparently iodine label is much better because it has lower in mice uptake in liver and gastrointestinal tract. So it was validated in clinics. In humans, unfortunately, not better at all. So we still had high liver uptake, though tumor could be visualized, as Olga has shown. Next step was selection of a right label or right format. So there were 2 different DARPins against HER2 selected, so-called 929 and J3. And J3 was somewhat smaller compared with 929 and had better affinity. And it had better affinity. Oh, sorry, I'm giving a lecture right now. Could you call me in half an hour? Apparently J3 was much better than 929, and we also looked at the way to reduce hepatic uptake. So we used 2 tags, hexahistidine and HEHE, and we tested if we placed at C terminus or at N terminus of the DARPin. So we found that if we place attack at N-terminus, we reduce hepatic uptake fourfold. And that might be the difference between success and failure. Right now, there is an ongoing clinical study in medical centrum, Tomsk Medical Centrum, where they evaluated this tracer in clinics. We got a very first real patient images, but we need to collect good statistics before we can state anything with security. We also continued to another molecular target, and this is EpCAM or EpCAM, pronounced in a different way. What is important? That this is so-called pancarcinoma antigen. It is very often overexpressed in many cancers, different cancers. For example, HER2 is breast cancer and gastroesophageal cancer, but that's it. Of course, to be able to treat those two cancers, it's good, but of course, you want to make a bigger impact on healthcare. EpCAM is considered as a cancer stem cell antigen also. So the most malignant cancer cell expressive. So it's, in our opinion, justifies development of therapeutics against EpCAM, and we also wanted to have companion diagnostics. So together with a team of academic dev, we developed a library of different EpCAM variants. We tested different approaches to labeling chemistry, and it seems that we are in a good way to develop. And we are working currently also on development of therapeutic probes which could deliver toxins or drugs to tumor using the same DARPin system. You might ask, well, you targeting HER2, you're using 3 different formats of scaffold: Affibody, DARPins, and that. Why? Isn't one enough? Well, if you like, it might be enough. But what is interesting here, that we have, for the moment, variety of different targeting agents against HER2, but majority of them bind to different epitope and even in different domains of HER2. So that means that you could use co-targeting. You can target the same target on cancer cells independently without interfering of each other binding. And that has a special value if you target for therapy. So efficient way is to have 2 different targeting agents which have a different toxicity profile, but which aims at the same molecular target in a cancer cell. In this way, you minimize the toxicity for each particular in normal tissue, but you focus the toxicity in tumor. And currently we are working on this co-targeting approach in radionuclide therapy, which is a completely separate story. And we intend to use both anti-HER2 and anti-EpCAM targeting probes to destroy a number of cancers, including breast, including ovarian carcinomas, for example. There is another interesting aspect. So different scaffolds have different preferences in their binding. For example, Affinity molecules have a flat binding surface, and that means that they need to have also flat antigen to be efficient. So if you don't have a flat antigen, Affinity molecules would not work. Though Affinity are my personal favorite, of course, but still. So we need different scaffolds to create a toolbox and select the best targeting agent for each particular target. And we have to remember that it's interdisciplinary, cross-disciplinary work which require multiple researchers with very different competence who manage to establish a common language and proceed from biotechnology to clinics. In our particular case, we have three major biotechnology partners. It's a company, Affinity AB, it's Royal Institute of Technology in Stockholm, it's Ovchinnikov Institute of Bioorganic Chemistry in Moscow. So we aggregate preclinical development in radiochemistry in Uppsala. We hope that soon it will be also done in Tomsk. And then we have a clinical partner at Uppsala University Hospital or Research Medical Center in Tomsk, where there are dedicated clinicians who are ready to work hard and fight difficult targets and establish a new protocol. We need to have a really good multidisciplinary team to be successful in this area. So, my take-home message from this lecture. So, radionuclide imaging helps make cancer treatment more personalized and therefore more efficient. So it helps to treat right patient with right therapeutics at right time moment. Smaller imaging probes improve both sensitivity and specificity of imaging because they provide higher contrast and lower unspecific uptake in tumor. The use of scaffold enables to develop small high-affinity probes to a variety of cancer-associated molecular targets. We're working with 8 different targets now, so it's possible. And we should always understand molecular design and optimal molecular design and optimal labeling chemistry to ensure that our targeting agent provides the highest sensitivity, specificity, and overall accuracy of molecular imaging. That's all for today, and we have a chance to ask questions, both in English or in Russian, as you wish. So you are very welcome.
No question.
No question. I hope it doesn't mean that everybody is sleeping. So that would mean that my lecture was too boring, which would hurt me. All right, exactly. No question at all. All right. Well, then, thank you. Спасибо за ваше внимание. Сегодня следующую лекцию будет давать Анжелика, и она просуммирует радионуклидную терапию. Это заметно отличающийся подход от имиджинга. То есть она требует немножко другой точки зрения. Ну ладно. Так, мне кто-то мелькнул в чате. Все. Нет, вопросов нет. Ну ладно, спасибо за внимание. Надеюсь встретиться через неделю. Через 2 точно встретимся.
Спасибо, всего доброго.
Спасибо
большое за лекцию.
До свидания.
До свидания.
До свидания.
До свидания.